So first we have Laurence Breville.
Since 2004, the yellow-legged hornet, Vespa velutina, spread into Europe and caused various impacts, mostly on insects. So to reduce this impact, a lot of technical methods have been deployed in order to destroy nests. And we wanted to destroy the nest using a thermal method. And in order to do that, we use 4 thermal methods at different temperatures. And the best method was the one with humid airflow, where all the hornet die only in 30 seconds. So the humid airflow was the deadliest to the hornets. So now we can use that technique in order to kill a hornet colony without pesticide. So now come and see my poster for more information.
Super, thank you so much. So our next speaker is Sonya Hakala, and she's from the University of Freiburg and also the University of Helsinki.
Can you hear me?
Yes.
Yes. Yes. So I'm from the University of Freiburg now, but the poster is about the work I did in Helsinki, doing my PhD on ant dispersal. And there are lots of ant species where young queens do not disperse, but instead stay in the original colony as extra queens. And these kind of societies have been studied a lot. And usually what we ask is, how is it beneficial for the whole society to have many queens? And in what circumstances that is? And of course, it often is beneficial, but that is not always the case. And our work focused on the other side of this story, the side where there is conflict over these dispersal decisions. And young queens try to stay in the colony against the will of the other colony members. So the poster asks a question of how to treat a daughter who refuses to leave the nest. So if you want to chat about complex dispersal conflicts in very complex social settings, come and talk to me.
Thank you, Sonya. Our next flash talk is from Malte Schulze. He is from the University of Copenhagen. So you can go ahead, Malte.
Can you hear me?
Yes, we can hear and see you.
Great. I'm interested in how the fungus Podaxis is adapted to overcome stress due to living in associations with termites. To test this, I have carried out grow assays on 9 different strains of Podaxis to quantify the effects of different hydroperoxide concentrations and to test for resistance to terpenes, which termites use in defense. At the moment, I'm analyzing the data from the project, and I hope the results will help to explain how the fungus can colonize and survive within the termite mound. Thank you very much for listening, and I hope to see you in the breakout room. Thanks.
Cool. Thanks, thanks, Malte. Next, we have Jason Rastin, and he's also from the University of Helsinki.
Yep, can you hear me?
Yes.
All right. So my poster is about my master's thesis, in which we tried to figure out how ants would self-medicate in nature and if aphid-stressed plants could be a source of medicine for them, because aphids can produce these systemic ROS responses in plants, and we know that ants can use ROS to medicate themselves in lab conditions. So we infected ants and gave them plants with aphids on them, and then looked at differences in foraging caused by this pathogen infection. And although we couldn't find any clear evidence that there's going to be ROS in this nectar, we did find that the ants are responding to this pathogen by increasing their foraging on the nectar during the first 3 days after infection, which fits both the infection cycle of the pathogen as well as a self-medication response. But we don't yet know how this change in behavior affects the survival of the colony. But if you have questions, you can find me in the breakout room.
Great, thank you so much. Very interesting. Next, we have Nils Pierboom, and he is from the University of Copenhagen, also discussing Podaxis termite interactions.
Yes, Hi, I'm a PhD student working on Podexus in Michael Paulsen's group. We can see the fungus growing from a grass harvesting termite mound in the back picture over here. Podexus exists as several predator species that segregate between a free-living, a facultative, and a specialist termite association lifestyle. This segregation has been confirmed by a recent global ITS phylogeny, and speciation will be confirmed by mating tests. Well, how often does this termite association occur? We sampled 52 mounds and we found the fungus on it in 17 cases, but this prevalence and impact on microbial diversity has to be confirmed by metabarcoding. And I will be happy to answer questions in the breakout room.
Great, thank you so much, Nils.